Hey, what's up, guys? This is Audie from Shastra Nation, and today we're going to be talking about the structure of DNA and sort of its discovery, I guess. So let's go ahead and get started. So the structure of DNA. Now, I'll explain the different parts of structure and then tell you guys how those were found out, basically, how this is going to work out. So really the main discovery, or at least the two people that the discovery of the structure of DNA was attributed to were Francis Crick, and James Watson. Now, these two didn't exactly find all of it by themselves. They did have help. They didn't have like direct help, but indirect help, and they made their decisions and certain tests and stuff like that based on the discoveries of other scientists around their time period or before their time period. So first, the main debate, I guess, at that time period was what was the actual genetic material? So was it protein or DNA? Now, this might seem a little bit silly to us, but if you think about it, it makes sense. Proteins have 20 amino acids, whereas DNA only has 4 nucleotides. And so to scientists that meant that there would be a lot more combinations of proteins than there were DNA, and so that meant that we would have more differences in the species and other organisms and stuff like that. So I guess if you think about it, it makes sense. Now, how they figured out that DNA was the actual genetic material is by they took out proteins from a different cell and placed it into another cell, and nothing happened, to the cell. Like, the cell didn't change parts or anything, like change what was happening inside the cell and things like that. But on the other hand, when they did put the DNA inside, the cell changed everything basically and became the cell that the DNA was taken from. It copied it exactly. And so then they found out that DNA is equal to the genetic material. Genetic material. So once they found that out, they wanted to figure out what the structure actually is. And so what they did is, they first thought that the nucleotides were on the outside of the DNA, and not on the inside as we do now. Now, I don't know why they thought that, but I guess it just made sense to them that it was like that. They didn't know that the nucleotides were bonded or anything like that. So then they found out that the nucleotides were on the inside, and now they had to figure out, what nucleotides bonded with what nucleotides. And so how they figured this out is by first they looked at the sizes. They looked at the sizes of the nucleotides. And they figured out, that A and G were big, or like C and T were big. I don't remember the exact sizes or anything like that. And that the other thing that these nucleotides base paired to were smaller. So now this wouldn't help them until they figured out that the DNA molecule was the same size all around. Now, what actually helped them the most is a discovery by Chargaff. Now, I don't know if that's exactly how you spell it, but you get the point. So what he figured out was he found out the ratios of the 4 types of nucleotides. So he found out that he found the ratios of the 4 types of nucleotides. And so by doing this, they could figure out what, by comparing the nucleotides of similar, the ratios of similar nucleotides, sorry, they could figure out what nucleotides paired together. And so when they did that, they found out that, adenine had the same ratio of base pairs to thymine, and then that guanine had the same ratio of base pairs to cytosine. And so by doing that, they figured out the, what nucleotides bonded together. And so this gave them an opportunity to figure out, that now the, size does matter, and that's probably why, the DNA molecule is the same all around. So this is how they found out what base pairs went together. So then they found out about the double helix by, actually Rosalind Franklin figured that out. So let's see, Rosalind Franklin. So Rosalind Franklin used this method called X-ray crystallography. X-ray crystallography is just basically like sending, taking an X-ray of a microscopic molecule or something like that. And so what she found was that that there is a high density in the thing like this. So it's like this, and by doing that, she figured out that DNA was in this spiral shape. So like that. That's pretty bad. But anyways, she found out that the DNA was in this spiral shape and that it was the same size all throughout. And so this supported this theory, basically, that the size of the nucleotides gave it, its shape and stuff like that. And so by using all of this, Watson and Crick first came up with how DNA was shaped, basically. So as I've told you already, it wasn't all of their idea, but they did piece together all of the information that was being found in the scientific world at that time. So when they did find out, there was it was on every magazine and everything like that, and even today, people still, if you go even if you go and ask like a kid in like first grade, don't know what DNA is. So DNA can be found in 2 different states, I guess. One in chromosomes. So we'll do chromosomes versus chromatin. So chromosomes are basically like tightly wound up DNA. So you might have like large strands of DNA that are tightly wound up with the use of histones, which are proteins. So histones, which are proteins that help do this, and they are tightly wound up. So why are they tightly wound up? Well, in mitosis, you need as much space as you can get, and so if they're tightly wound up, they're easier to they'll be replicated, and then they're going to be tied up really tightly, and they can be separated and moved apart with ease. Now chromatin. Now the problem with chromosomes being there all the time is the fact that it's really difficult to actually use the DNA. So we need the DNA to make proteins and enzymes and other things like that. And so if the DNA isn't usable because it's so tightly wound up, then you can't use it, and therefore, you won't get anything done. And so chromatin is the form where it's sort of loosely bound. and easier to replicate. So it's loosely bound and easier to replicate, meaning that it can be used by the cell to make proteins and do whatever else it needs. So yeah, also, let's go ahead and talk about the structure of a chromosome. So a chromosome looks like this. You have this, and you have this. So, really it should be like this. This is not an actual drawing, actually. So, let me go ahead and erase that. Alright. So it should really look like this, where it's like this and this. And here would be the thing that's holding it together, or as scientists call it, the centromere. So the centromere is basically the thing that holds the two sister chromatids. So these things are called sister chromatids. And so why they're called sister chromatids is because they hold the exact same DNA. So if we took out this part and compared them, they would have the same DNA on both sides. Or if we took out this whole part and compared it to this whole part, it would have the same DNA. And so that's what this is why, you know, once they're replicated and they break apart, so this side will go this way and this side might go this way. And once that's done, you know, the two cells can use the exact same DNA because of mitosis and meiosis. Mainly mitosis. So yeah, also the DNA, we talked about the, you know, we talked about the double helix, you know, model. That is not a good picture of that. I will redraw that. Okay, so we talked about the double helix model, right? Now I'm not going to just leave you at that, but here, so these might be the nucleotides. These ladder rungs, I guess they're called. These are going to be the nucleotides, you know, and they're going to run all throughout the DNA molecule like this. Now, the thing in the back that's holding it is called the backbone, and the DNA backbone is made up of a series of sugars and phosphate bonds. So sugar, phosphate, in any order. And so basically, these things hold together the molecule, and, you know, this bonds to this, this bonds to this, this bonds to this, you know, and it just keeps going on until it ends, basically. Now, also, we have this thing called the 3' end and the 5' end. So it runs an opposite. So it's what they actually call this is the anti-parallel, and so it runs in opposite ways. So this might be the 3' end, and that's how you write it. And this side might be the this side would be the 5' end if that was the 3' end, you know. And then this would be the 5' end, and then this would be the 3' end. And so it always has to go that way. So then, you know, we'll talk about more on how this, you know, matters and how things can change, you know, how this changes things and stuff like that when we talk about transcription and translation and other things like that. But basically, all you have to know for this section is some of the people that came up with it, you know, and what they came up with, how that was used to come up with the overall structure of the DNA, and who actually came up with the DNA, and why DNA is considered, you know, the history of DNA versus protein as the genetic material. Also, the structure of a chromosome, the difference between chromosomes and chromatin, and just stuff like that. So anyways, guys, I hope this helped. If you like my videos, go ahead and hit the subscribe button and share it with a couple of friends. Also, if you liked my video, hit the like button, share it with a couple of friends. I come out with videos like this almost every week, every Monday actually. And if you have any questions, let me know in the comments below. Or if you have any suggestions, go and click on the suggestion link in the description, And I guess I'll see you guys in the next one.